Dear delegates, we are starting with the next session soon.
Good evening, everyone. स्पीच पहले देख ले फिर ऑनलाइन हो।
Due to some technical problem, Rajiv Kumar sir was not able to join this session. So we are moving forward for our today's first scientific talk. And for the same, we have the chairperson, Mr. Rajiv Kumar, with us. And it gives me an immense pleasure to introduce the esteemed and renowned personality on this occasion. Mr. Rajiv Kumar is working as a head manufacturing at Biologics division of Dr. Reddy's Laboratories, Hyderabad. Sir is a postgraduate in industrial microbiology and is having 21 years of experience in biotechnology industry. He has worked on different biotechnology-based products, including monoclonal antibodies, recombinant therapeutic proteins, vaccines, and antibiotics. And Sir has keen interest and expertise in aseptic manufacturing of aforesaid biotechnology drug products. We are highly obliged to have you, sir, today here, and we extend a hearty welcome to you. Now, we are also fortunate to have with us Dr. Kapil Maithilsar as a first speaker of our session. And it's give me an immense pleasure to introduce him. Dr. Kapil Maithil is currently working as a president, vaccines and diagnostic at Zydus Cadila, India. Sir has over 2 decades of experience in developing and manufacturing of vaccines, biologicals, and diagnostic from concept to launch. He is trained as a protein chemist and a structural biologist and has been in the field of vaccinology for over 2 decades. Sir has worked in academia, industry, as well as PDP organizations in various roles. Sir is also interested in global public health and served as a committee member of Independent Review Committee of Carry Alliance for the number of years. He has also number of patents and international publications to his credit and has been active at Global Forum and is a member of number of academic as well as scientific committees. We are highly obliged to have you here today, sir, and we extend our hearty welcome to you, sir. And may I now invite Dr. Kapil Maithil, sir, to deliver his talk on vaccine developments for the COVID-19. Over to you, sir.
Thank you so much for the kind introduction. Are my slides visible?
Yes, sir. Very good. Pretty much visible.
Yeah. So I thought the current hot topic about COVID and the COVID vaccines which are in development would be an area of interest to the participants. So that is what I'll talk about. And the slide basically would cover some basics about the coronavirus disease, 2019, in short form called COVID-19, vaccines in general, and then finally we'll talk about COVID vaccines. So I think it is important to know that pandemics have been with the mankind for centuries. There have been a lot of cases which have been reported in the past where the pandemics have rocked the history of mankind. And some of the major ones have been captured here, but there have been so many more. The first one is bubonic plague, which had hit in the 14th century, leading to almost 200 million deaths. It is also called as Black Death, one of the largest pandemic which had impacted the mankind. And in fact, when I say the history of mankind at that point of time, there was a war which was going between England and France, and they called truce because none of them were able to manage with such a vast pandemic which had hit their economies at that point of time. In fact, the British feudal system had totally collapsed because of this. The second one, which I'll mention, is about smallpox, which came somewhere in 1520, leading to 56 million deaths, which had happened at that point of time. And they say that the Aztec Empire, which was very prevalent and very dominant in Latin America near Mexico, in fact, was completely collapsed. The empire collapsed because of smallpox. In the last century, Spanish flu was one of the major pandemic which had hit, and this was similar to the swine flu pandemic, which happened in 2009-10 globally. So it was also caused by H1N1 virus, and at that point of time, there was no vaccine or antiviral which was available to control it, leading to almost again 50 million deaths globally. HIV is another pandemic which still continues. In 1918, it came and gradually it has increased, leading to almost 35 million deaths so far, and we are still struggling to have proper vaccine for it. There are certain antiretrovirals which are available to control, but obviously doesn't eradicate the virus completely. The recent one is the COVID-19, which has hit us, and already there are around 2.28 million deaths which have happened globally. So these pandemics have been with us, they keep on coming, and it is how well we gear up and address this pandemic is what is important for us. So what is coronavirus? So coronavirus is a large group of viruses, and there have been earlier incidences of coronaviral infections. In dogs, there is already a coronavirus vaccine, but it is for a different class of coronavirus. And the major ones in humans was the SARS-CoV-1, which had come in 2002 to 2004 in China, but again, it was controlled well and did not spread much at that point of time. Then came the MERS coronavirus, which is Middle East Respiratory Syndrome in Jeddah in Saudi Arabia, but again, the cases were controlled. It did cross around 26, 27 countries, but the number of cases were low, so we were able to manage, unlike the current pandemic which is going on. Looking at the virus, you will see these electron microscopic images courtesy of CDC, and these are the spikes on the surface of the virus due to which it gets the name, looks like a crown, so they are called coronaviruses. These are the different proteins on the left-hand side, lower left-hand side, you see, which are associated with the virus. The major target for most of the vaccine is the spike glycoprotein S, which basically is a component of these crown-like structures protruding out of the virus. The virus itself is around 30 kilo base pair, and this is the assembly of the virus. And here, the spike protein is one of the large sequences, as you would see, besides ORF1A and ORF1B, which is, as I mentioned, a target for most of the vaccine candidates. Coming to some of the statistics of how the incidence of this disease has happened. So in early March, as you would see, when the incidence started happening, so most of the European countries started getting impacted after the cases in China were regressing, and suddenly then it came to the Latin America, North America, where the cases started increasing. In India, the cases started increasing somewhere in June. They peaked in September, and ever since it has been declining. And again, recently in November, you would have heard about the resurgence seen in Europe, which was led by UK, the new variant which emerged, and followed by South Africa, Brazil, and again in US, you see the cases have gradually started increasing, primarily because of the mutated virus or relaxation in public health measures. Now, unlike other pandemics, here the older adults are at a greater risk. 8 out of the 10 COVID-19 deaths in US, this is a figure which I have taken from CDC site, and you would see that the major number of deaths have been associated in elderly population, and that numbers gradually come down. And in fact, you would have heard not many cases in the pediatric population or any mortality which has been observed in the pediatric population. So there is a lot of risk in the elderly, and they are also transmitting the virus. So a lot of precautions and safety measures have to be taken care. So how does coronavirus impact the body? So I think most of you would know about this. So it is day 1 to 3, the onset of symptoms post incubation may start with upper respiratory symptoms, fever, cough, cold. 80% of the cases are at this stage. The cases which further progress to the disease, day 4 to 9, is in the lungs, and the disease may reach in lungs between 3 to 4 days. There is difficulty in breathing, inflammation, followed by ARDS in the lungs, and 14% of those infected experience these kind of severe symptoms. Some of the cases which further progress are on day 8 to 15, and here the virus enters the blood, can lead to sepsis, and 5% of those infected need ICU care. And this is very difficult phase for these patients, and many of them are not able to survive. And usually the period from infection to cure is around 3 weeks. And there are also post-COVID complications. And some of you would have heard from your friends or relatives who got infected that even after cure, there are a lot of other complications. There is a lot of lethargy. There are a lot of other comorbid conditions which had emerged in their bodies, taking a longer time to recover. They call it as long COVID scientifically these days. Coming to vaccines per se, so there are 8 distinct vaccine market segments. So they are traditional pediatric vaccines, which most of you would have heard about, like a pentavalent vaccine, which contains diphtheria, pertussis, tetanus, Haemophilus influenza type B, as well as hepatitis B, or a DPT class of vaccine, polio, rotavirus, MMR, measles, rubella, tetanus. So these are common traditional pediatric vaccines made by available technologies. Strongly regulated pricing for these vaccines, since most of them are part of EPI. Specialty pediatric populations is the vaccines which are now the new vaccines which are emerging with innovative technologies like the pneumococcal, meningococcal, or RSV vaccines. Pneumococcal this year in the budget, the finance minister has said would be introduced in the EPI program, pneumococcal conjugate vaccine for pediatric use. So another vaccine getting into EPI, a very good vaccine to vaccinate the kids. A lot of mortality would be controlled because of it. Then there are adult booster vaccines, which are given primarily to adolescents and adults to refresh and strengthen the protection by primary childhood vaccination, like TD, which is tetanus and diphtheria, or Tdap, which is tetanus, diphtheria, and acellular pertussis, which is a booster which is usually given 10 years after the vaccination period. Similarly, for a pneumococcal vaccine. Then there are specialty adolescent adult vaccines, which are newly targeted infectious diseases with high unmet need. HPV vaccine, the human papillomavirus vaccine, which basically causes cervical cancer. Already in some states, this vaccine is being introduced in India. Then pneumococcal vaccine, herpes zoster vaccine, rabies vaccine, which is more to do with post-exposure prophylaxis rather than pre-exposure prophylaxis, where it is being used. So the people who are bitten by dog are given the rabies vaccines along with immunoglobulins for protection. It's a very dangerous disease. So once it enters the neural tissues, no one can protect an individual. After a dog bite, mortality is certain. So a very important vaccine. Then there are other vaccines which are used as travelers or endemic vaccines. This is for the vaccines which are contained in particular territories, and usually travelers or military people take these kind of vaccines. Hepatitis C, meningococcal A vaccine, or a yellow fever vaccines. Many of you who would have traveled to Latin America or certain countries in Africa would have taken a yellow fever vaccine as a mandatory requirement. Seasonal or pandemic vaccines, influenza is a classic case where, because of the variability or shift and drift in the strain of the influenza, there are new strains which emerge, and every year one needs to take a vaccine for that. Anthrax, now COVID-19 is a pandemic vaccine which has hit. Anthrax is a vaccine, around the time of bioterrorism, there was a big scare about the anthrax, and anthrax vaccines became available and very popular at that point of time. Usually this vaccine is available for ruminants, but in humans now the vaccines are available. Then there are certain therapeutic vaccines, and this is leverages to treat diseases such as cancer, oncology. There is a vaccine already approved for prostate cancer. Then there are vaccines which are in development for Alzheimer's, drug addiction, allergy, and then there are vaccines for special population, primarily for hospitalized population. Staph aureus is one of the major causes of sepsis in hospitalized population. Another important vaccine under development, no vaccine is available as of now, is for Clostridium difficile, which is a cause for diarrhea, and again a disease leading to high mortality rates as of now. Here you see a worm of the vaccine development, and you see the first vaccine was developed for smallpox in 1798 by Edward Jenner, also referred to as the father of vaccinology, and he developed a vaccine where he used a cowpox lysate, or the pus formed from the cowpox infection, which was not leading to mortality. And that material he used to vaccinate the son of a gardener, his own gardener. He vaccinated the son of his own gardener, and he found during a challenge that the disease did not happen. As I had mentioned earlier, there was a lot of deaths associated with smallpox before that, and it's turned out to be a miracle. And Edward Jenner got a lot of recognition also in UK at that point of time. Then almost it took 100 years before the second class of vaccine came in, typhoid and a rabies vaccine, followed by, in 1920s, a lot of toxoid vaccines started coming in, which like diphtheria toxoid or tetanus toxoid, and then gradually the field picked up and more vaccines started emerging. And here you would see from 1910 to 210, a lot of vaccines were developed. The polio vaccine came somewhere in 1963, I believe. Yeah. So the oral polio vaccines, which many of you would have seen as a part of the pulse polio program in the country, came in 1963. Then the rotavirus vaccine came in 1999. Then the pneumococcal conjugate vaccine, the first vaccine, came in 2010. Then there have been a lot of vaccines. So here I have not covered the last decade, but there have been a lot of vaccines which had come. In 2014, there was a vaccine for Group B meningococcal, again a very good vaccine, which was developed by Pfizer by the name Trumemba. Then there was, in 2015, the first malaria vaccine, Mosquirix of GSK, which got an approval with recommendations. And in 2020, we also got a vaccine for Ebola. So we all were struggling with the COVID pandemic, but there was a good news in getting a vaccine for Ebola. which had caused a lot of deaths in African region. So these are some of the new candidates for vaccine which are in development, and on the x-axis you will see from lower potential impact to a higher potential impact in terms of mortality and the lives which would be saved once the vaccines are introduced. So on the right hand corner you will see the diseases like HIV, tuberculosis, hepatitis C. We don't have any good vaccines available for these diseases. Malaria, we have a vaccine, but again, still it is under recommendation which has been approved. Then there is non-typable Haemophilus influenza, again, infection leading to high mortality annually. Then there are certain other diseases, and on the left-hand corner you'll see diseases like seasonal influenza, maternal influenza, meningitis, for which the vaccines are available, are getting introduced in most of the countries. Now, just to give you an idea about the time scale for new vaccine development, now many of you would have heard about the rapid pace with which the COVID-19 vaccines have been developed, and the people have some sort of an apprehension. And why is that? Because as you would see, most of the vaccines take years to develop. And for something like HIV, still 37 years had elapsed since the virus was discovered, and still we don't have any good vaccine available. Malaria took 122 years to get a vaccine. Typhoid, more than 100 years. Today, typhoid vaccines are so commonly used, but it took 100 years to get a vaccine for that. Whereas for COVID-19, it was less than 1 year. The first vaccine got approved in Russia in the Q3 of 2020. Followed by Pfizer getting approval in December of last year. If you now look at the impact of vaccines on global health, this is different diseases. There was a large number of deaths. This is data of UK, which I have captured here. And so you see that once the vaccine was introduced for diphtheria in 1940, the number of cases came down drastically. Almost no cases was observed. Similarly, in meningococcal infection, cases just went down to zero. Measles, which is basically an episodic infection, but again, the cases came down once the vaccines was introduced. And similarly for polio, Hib, pertussis, the cases significantly came down. You do see some peaks coming in Hib in 2002, and also for pertussis, primarily because of low vaccination rate in the biblical belt in UK, where the cases increased because of certain ethnic populations that were not vaccinating their kids. As a result, the infection was found in these years. But you see the impact the vaccines have had on the human health. Now, coming to the COVID vaccines, you see after the virus genome sequence was available in January of 2020, there had been a spurt in the vaccine development. Globally, so many players were developing. Even we at Zydus have developed 2 vaccine candidates, and I'll talk a bit about them a little later. So currently, this is as of 3rd February from New York Times COVID-19 vaccine tracker. So there are 36 candidates in phase 1 clinical trials globally, 25 in phase 2, 20 in phase 3, 7 have got limited use authorization, including the 2 vaccines in our country which has got emergency use authorization, Covishield and Covaxin. And there are 3 vaccines which have been approved, full licensure, and there have been candidates which have been abandoned. There are 4 vaccines which have been abandoned, 2 coming from Merck, 1 coming from University of Queensland in Australia, and another one coming from Robin Shattock's lab in Imperial College. So there have been different classes of vaccines which people have worked on. There have been novel approaches which people have looked into. So there are protein subunit vaccines. Then there are viral vector vaccines. Now in viral vector, you have both replicating and non-replicating viral vaccines which have been developed. There are DNA vaccines which have come up, inactivated virus vaccines, the conventional vaccine types, or an mRNA-based vaccines, the likes of Pfizer and Moderna, or virus-like particle vaccines similar to what Novavax has developed. And then there are live attenuated viruses where codon deoptimized viruses are being used. And I'll talk again about that in my coming slides. So these are some of the vaccine platforms which have been utilized for the COVID vaccine development. And you would see the first one is a protein subunit vaccine. Examples are Novavax, COVAX. COVAX is a vaccine candidate which is now being, Aurobindo Pharma in India is working very closely with them, and they would be conducting clinical trials in India. Biological Evans candidate is a subunit vaccine, and they are also getting into clinical trials. They are already in clinical trials. And these are the fragments of proteins or protein shells that mimic the coronavirus outer coat to trigger an immune response and stimulate immunity. The advantages are they are non-infectious, manufacturing is not stringent, but they may require adjuvant. And if there are novel adjuvants which are being used, you would need to generate a lot of data on that. And as we hear about the COVID-19 infection, there is a need of both the arms of immune protection, humoral as well as cellular, and many of the conventional adjuvants, which are aluminum-based, do not confer a cellular protection. So they are more Th2 biased responses, not Th1 biased, which is believed to provide better protection for the COVID vaccines. Coming to the second category of vaccines are viral vector-based vaccines. As I mentioned, that there are two categories: non-replicating viral vaccines, which are primarily based on adenoviral platform, which is currently in development, likes of Sputnik, the Russian vaccine from Gamaleya Institute. In India, DRL is working with them, getting into clinical trials. Then there is an Oxford AstraZeneca vaccine, which is on chip adenoviral platform. And in India, Serum is working on that. And Covishield is the vaccine you have all heard about. Then there is a Johnson & Johnson, again, an human adenoviral-based vaccine. And they just published their data in Lancet, and the results are very good for their vaccine as well. The single dose is providing good protection, followed by the booster dose. Then we are also working on a replicating viral vaccines, which is based on a measles platform. The measles, as you know, is the vaccine is being given to billions of people. So we believe that it would be a good vaccine to work on. These vaccines give both good cellular and humoral response. That's the hallmark of these vaccines. The only challenge is that immunity against the vector which carries the antigen gene may be formed. So if, for example, in adenoviral-based vaccines, you get a lot of antibodies to the adenovirus, the booster shots may not give that kind of a protection. So that is a kind of a challenge which these vaccines possess. Again, these are early days. There is already some data showing that it may not happen, and we may be able to give a lot of boosters for these vaccines. High dose of these vaccines are required, but as the field has evolved, people have been able to develop a lot of good protection for these vaccines, processes for these vaccines to prepare high dose. Then there are DNA-based vaccines. As I mentioned, at Zydus, we are working on these vaccines. So our vaccine is now in phase 3 clinical trial. A US company called Inovio is also working on this vaccine, and they are also into phase 2-3 clinical trials as of now. And there are a number of other DNA-based vaccines which are in clinical and preclinical development, which are moving fast. The advantage of DNA-based vaccines is that they are highly stable vaccines. They can be stored at room temperature for prolonged periods. Again, the platform is very simple. You can adapt quickly to the new viral strains if the mutation is observed. But the disadvantage is that it may require specialized devices for efficient uptake of the vaccine candidate, uh, rather than simply giving it by a normal needle and syringe. The last category is mRNA-based vaccines. So these are again a nucleic acid-based vaccine, uh, and you have all heard about Pfizer and Moderna. Another good candidate from CureVac is now in advanced clinical trials, and you would hear more about it in coming weeks or by next month. The advantage is that again it can be very rapidly developed, uh, and can be adapted quickly with the new mutations. The only disadvantage currently is that it requires sub-zero storage conditions. There are a lot of side effects which have been observed with RNA-based vaccines. You would have heard about the lot of deaths which happened in elderly population in Norway. There have been Bell's palsy associated with it. So there are certain side effects, but the efficacy data is very good. And I'll show you a slide on that as well as we move forward. Other classes are the inactivated viral vaccines. So this is the conventional vaccines. So Bharat Biotech in India, along with ICMR, has developed this vaccine called Covaxin, which is based on this platform. Sinovac in China, another company, Sinopharm, which is the government-owned another company in China, have also worked on the same platform. They use certain chemicals like formaldehyde or beta-propiolactone for inactivation of the virus, and it generates a good immune response. Again, the disadvantage is that it may need adjuvant. Fortunately, the Bharat Biotech vaccine uses an imicodazole adjuvant along with their aluminum adjuvant, which confers a very good Th1 as well as Th2 response. They have recently published their phase 1 clinical trial in Lancet. Some of you may want to read that. The only challenge here is that they work on the wild type virus, which requires stringent manufacturing facility requirements, which is biosafety level 3, and scale can be a challenge. The containment issues would be there for the waste generated at these facilities. So maintenance is high. So that is the downside of such platforms. Then there are live attenuated viral vaccines. These are codon deoptimized vaccines. So Serum and Indian Immunologicals are working in India on this kind of platform. And these are weakened viruses. So you would have heard about codon optimization when you do expression studies. So what here it is done is they do a de-optimization of the viral genome so that it doesn't replicate to the extent which can cause disease, and the immune system is able to confer protection by the time the virus starts replicating. Then there is the advantage is that there is again a very good immune response because we're talking about the whole virus. Adaptation to new virus strain is not quick. That is the downside. There is also a risk of reversion in such candidates because if the virus mutates and reverts back to a more virulent form, it may not provide protection. Again, it would require stringent manufacturing facility of biosafety level 3. The last category is like virus-like particle, which I mentioned. Medicago is working on this candidate. The advantage is it is again non-infectious. Multivalent formulation can be made with ease. Several proteins need to be produced simultaneously for VLP vaccines, and manufacturing is not easy. So that is the downside. of such platforms, you need to ascertain that the virus-like particles are formed well from batch to batch. So you need to have that kind of consistency. So process development takes time. If the strain changes, the new construct will need to go through the entire process development path. So that could be a challenge. Just to refresh about the rigorous clinical trial evaluation which the vaccines go, there have been some concerns in the recent times about the clinical trials for COVID vaccine. But let me assure you, the extensive work the manufacturers, regulators, and clinicians have done on these vaccines will ensure that most of the candidates would be safe before it reaches the masses. So there are 4 phases of the clinical trial. This is the conventional phases. Phase 1 determines the safety primarily, as well as dose determination. Phase 2, again, one looks for safety and immunogenicity. Phase 3 is where the efficacy is done. It's a long trial in large number of subjects. And phase 4 trial is actually post-marketing surveillance, where the durability of the immune response and protection and safety is evaluated when the vaccine is given in the large number of population. You would hear about different grades of adverse events which are associated with vaccines. Grade 1 is mild, which does not interfere with routine activity like cough or mild fever or rash at the site of injection, redness. Grade 2 is moderate, and they may interfere with daily activities like fever and severe pain, malaise. Grade 3 is severe, which prevents daily activity and requires OPD visit. One needs to go to a doctor. The symptoms are so severe, there could be convulsions. And life-threatening, where it requires ER visit and hospitalization. These are some of the symptoms or adverse events which have been associated with COVID vaccines, including redness at the site of injection, swelling, fever, malaise, vomiting. There was a case associated with myelin sheath getting impacted for the Oxford vaccine, then Guillain-Barré syndrome, Bell's palsy. These are the different symptoms which have been, or adverse events which have been associated with the current vaccines. Now, just to give you an example of the adverse events associated with vaccines in various clinical trials, and some of these vaccines you would note are the ones which has quite commonly been administered and taken by us. Even the kids get vaccination. And there have been cases like the local reactions associated with pain, swelling, and redness. BCG has more than 90% of that. DPT has up to 50%. So it's not that only the COVID vaccines have these kind of reactions. Most of the vaccines do have these kind of reactions, but they resolve very fast. I think that is important. If there is something which is continuing and leading to a serious adverse effect, I think that is what is worrying. So I've listed down some of the serious adverse events associated with some of the vaccines. And for COVID-19, autoimmune disease, severe pain and swelling, anaphylaxis. Primarily, you would have heard about the people having some allergies, having an anaphylactic shock with Pfizer vaccine in some of the countries in US and Europe. So these are the cases which are associated with these vaccines, but they resolve fast. I think that is what is important for the current approved vaccines. So this is a comparison of some of the approved vaccines. So you would see the efficacy for the 2 mRNA vaccine ranges from 95% to 94%. Sputnik just published their phase 3 clinical data, and it showed an efficacy of 91.6%. These are excellent efficacies compared to some of the other vaccines which are available. The flu vaccine, for example, has an efficacy of 50 to 70%. Most of the vaccines don't have that high efficacy. So an AstraZeneca vaccine having a 70% efficacy with the 2 full doses is a good efficacy. Obviously not as good as the other 3 vaccines, but it would be considered good as long as it has a better safety profile. The Sinovac vaccine, again, they recently Brazilian data came out and it gave a very modest, or I would say a weak efficacy of 50.4. WHO has recommended that the vaccine should have more than 50% efficacy to move forward, and they were just at border. But the trial in China had given an efficacy of 78%. So this basically highlights the importance of different races, different ethnic origin, as well as geographies where the vaccines needs to be tested before it goes into humans, because you may get different kind of efficacy data which would be very important to a certain. Similarly, safety would also depend on the kind of population which is being given these kind of vaccines. Adverse events, you would see there are high number of cases which have been listed here. Serious adverse events are around 2% to 2.9% for the 2 mRNA vaccine. AstraZeneca has a lesser 1% cases. Sputnik has reported very low 0.3% of the cases. Then the storage condition for the mRNA vaccine is at minus 70 degrees Celsius. Moderna claims that at 2 to 8, it would be stable for 2 to 8 degrees Celsius for 30 days, but the storage condition, the real-time condition is minus 20 degrees Celsius for the Moderna vaccine as well. AstraZeneca has currently reported a 6-month stability for 2 to 8 degrees Celsius storage. The Russian Sputnik, they have 2 formulations. One is in liquid formulation, which is at minus 18 degrees Celsius, and the lyophilized formulation is stable at 2 to 8 degrees Celsius. The Sinovac, the inactivated vaccine, is stable at 2 to 8 degrees Celsius. Now, this storage conditions becomes very important for countries like us and other LMICs, where the distribution of vaccine is very critical to reach the masses, where maintaining such kind of cold chain can be a challenge. So, yeah. Just giving you a glimpse about the emergence of new mutants, and there has been a worry globally that how will these vaccines work. This is a slide which I've just taken from a recent workshop which was organized by the COVAX group, and talks about the 3 variants, the UK variant, the South African variant, and the Brazilian variant. And all these 3 variants have this mutation N501Y. And this is a region which enhances the binding affinity to the ACE2 receptor, the primary target of the spike protein, and it increases infectivity. So that is why you would have heard that these strains are highly infectious. Fortunately for the UK variant, the current vaccines are providing good protection, equivalent protection to the Wuhan strain, the conventional strain which was identified in China. But for the other 2, that's not true. There is a diminished protection, significantly reduced protection, and the convalescent sera and the monoclonal antibodies targeting of class 1 and class 2 type, which are targeting E484K mutation or a lysine 417 asparagine mutation are not protective. So the antibodies from Lilly, which is giving a very good effectiveness, may not work once this variant comes in. So is the case with the Regeneron monoclonal antibody cocktail, where one antibody is giving protection against the South African variant. The second one is not. So I think this is, as the escape mutants will start forming, one would need to keep a lookout and developing vaccine candidates which can provide or confer more protection. One may need to design vaccines at more conserved regions or the structures which would be able to provide protection. The flip side is that since there would be number of mutants which gradually will emerge, so a vaccine which is, say, developed against a South African variant, will it provide protection to the conventional Wuhan strain? Now, that kind of data is not available as of now, but having said that, one may need a cocktail of antibodies or a cocktail of antigens as a part of a vaccine presentation, which will one need to look at. Just to give you a brief about the vaccines which we are working on. So we are primarily focused when we started working on the COVID vaccine development somewhere in February, March of 2020 was safety. And we chose two platforms. One is on plasmid DNA vaccine, and the second is on a measles vector vaccine platform, which I mentioned earlier to you. And DNA vaccines have been proven to be very safe. There is a data of over 35,000 people without a single major adverse event. We have found this vaccine to be very safe in a phase 1 and phase 2 clinical trials. In fact, the initial vaccines which were developed for coronavirus infection for SARS-1 and MERS were DNA vaccines which had advanced to later stages of the clinical trial. But as the disease was controlled, these candidates did not move to a larger phase 3 clinical trials. The second candidate is a measles vectored vaccines. So here the spike gene is engineered on the surface of the measles virus, and it's a replicating vector vaccine. So you do see a replication in the host. So one does not need to administer a very high doses what one would need to give in a non-replicating platform like adenoviral vaccines. You would have heard about 10 to the power 10 virus particles which are given for non-replicating viral vaccines, whereas for measles vector vaccine one can go to 10 to the power 5 or 6 at the most. The platform is very safe. The only challenge or risk associated which people have argued is about the antibodies already persisting to measles in the population. But there is a beautiful article published by Themis where they have done a large clinical trial for a chikungunya vaccines based on the same platform, and they have shown that persisting antibodies to measles do not interfere and provide protection to the chikungunya vaccine. Similar data has been published by other academic groups, and these kind of vaccines will serve as bivalent vaccines and confer a booster to the measles disease as well as to the COVID infection. Just talking about our DNA-based vaccine, some of the merits. So they are non-infectious, they have no anti-vector immunity risk. There is no adjuvant which is required. DNA vaccines are known to induce a very strong humoral and cellular responses, which we have also seen with our vaccine candidate. And they can be rapidly scaled up. The manufacturing requirements are biosafety level 1, so one can scale up or go to any CMO and produce it if need be arises to have large number of doses. And in case the virus mutates, as we are seeing now, it is only a couple of weeks where the new candidate can be generated, and the process would remain same. So usually there is no change in the manufacturing process, which makes it very easy platform to work upon. This is just a comparison on the lower half of the screen, talking about the safety and efficacy for the groups. I will not go into detail as I've already covered part of it in my previous slides. So the major advantage is what we have seen for our vaccine is that it is stable for 3 months at 25 degrees Celsius, confers a strong immune response in animals and non-humans. We have found it to provide protective efficacy in a non-human primate challenge model as well. The wild-type virus is completely neutralized by the sera generated by a vaccine. There are no safety concerns. The vaccine is currently into phase 3 clinical trials, which we expect to complete by end of first quarter or early quarter 2 of 2021. So that is my last slide. Thank you so much.
Thanks, Dr. Maithil, for bringing this all very elaborate information on the COVID vaccine scenario. So I have few questions which is coming through the platform. What is the approach which Zydus has taken for fixing the dosage of the vaccine, and how the challenge is being taken up in line to what learnings we have in the industry so far?
Thank you, Dr. Rajiv. So talking about the dose selection, so we have done a lot of animal data, and based on the animal data, a dose was selected for the vaccine candidate. One would also need to look at the kind of feasibility of administration of the vaccine, and that was also a criteria which was checked. So a maximum dose what we have checked in the clinical trial is at 2 milligram, but we did demonstrate up to 6 milligram safety in an animal tox study. So yes, we could go a higher dose, but at present the trial was done at a 2 milligram dose. What was your second question?
Yeah, the second question which I'm asking is, what are the learnings which we have as an industry, as a WHO pandemic surveillance program, as a government? What you can summarize? What are the big learnings which can be implemented and institutionalized, make it systemic changes, so that mankind can take care of such kind of pandemic in future? So being a scientist, being so much behind vaccines in your life, what are the key takeaways from the current learning, current pandemic, so that the systemic changes can be suggested to the competent authorities?
So I think a lot of you would have heard about the initiatives which have been taken in the current budget. There would be a lot of institutes which would be working on viral diseases and viral infections. I think that is very important. Currently, there are limited number of institutes, only a premier institute like National Institute of Virology in Pune, which is leading these kind of activities. But I think more institutes would be required when the pandemic of the current scale happens, and with the new mutants which would be emerging. I think the second most important initiative would be on genomic sequencing. Again, already the government has taken initiative and identified few centers in the country where the sequencing of the virus can happen, which is again be very important, because one knows when it's an RNA virus infection, there is bound to be mutations which would happen. The third thing which is very important is improving the surveillance system in the country. I think that is an area which has been lacking, but again, I think this pandemic has brought to fore that it's very important to know the pockets and territories, how the transmission of a disease is happening, what kind of community transmission would happen, what kind of a herd immunity would be developed because of this infection, and what kind of interventions one needs to take in selective pockets. India being a vast country, it's not that what would be true for the western part of the country would hold true for the eastern part of the country, or a northern and a southern. So there would be different rates of infections. Climate is very different in a country in different territories. And I think One Health is another important initiative which is being talked about and needs to be implemented, because most of the 60% of the diseases are zoonotic in nature.
Yes.
So needs to develop interventions to not only work on human diseases but also on veterinary diseases. Because even this COVID, still the source of the animal source of the is still there is a lot of questions on that, but probably bats transmitted it to some species from which it came to humans. So I think that kind of interventions is very important. Now, as I showed in one of my slides, that there is a deletion which has happened for this virus in the mink population. There was a lot of culling you would have heard about in Denmark and other countries of mink. I think these mutants would cross over to people who had this disease. So I think one would need to work on multiple aspects, and I think SEPI and Gavi were already prepared. They were calling it as a disease X, which is going to hit the mankind. I think this is the disease X which is hit, and there were a lot of interventions done. But yes, I think these are some of the areas one will need to focus on. I think we lost Dr. Rajiv.
Maybe due to some technical reasons, sir may have left the meeting. I think he has joined. So we are very thankful to Dr. Kapil Maithilsar for such a delightful talk and being a part of the event and sharing your views as well as expertise with us. Once again, we extend our thanks as well as gratitude to you, sir, for sharing the valuable knowledge with us.
Thank you so much, and have a good conference. Bye.
Yeah, thank you so much.
So we may wait for sir to join back. Yes,
Palak, show the
yes,
certificate.
madam. But Kapil sir has also left the meeting, so we wait for him
Yeah,
1 or 2 minutes
but
and will
yeah,
present the certificate. So, on behalf of LM College of Pharmacy as well as the uh, our team, we are very thankful to Dr. Kapil Maithal sir as well as Dr. Rajiv Kumar sir for sharing their valuable time with us as well as uh, so gracing this occasion. Now, I invite Dr. Preeti Verma, lecturer, LM College of Pharmacy, to coordinate the next session. Over to you, Preeti madam.
Thank you, Dr. Palak, and a very good evening to all of you. I again extend a hearty and warm welcome to all the participants and guests in this second day of international conference. Before we proceed to the further session, we would like to wait for our chairperson and the speaker to join. So we'll be continuing with the session soon. By the time, we'll be just displaying the display advertisement here. So be with us.
Start every day. Help move science forward. So we can all live your life on a careful, your wall. This is why we work. To help solve, to save, to advance chemistry. Because when we partner with science, we make the future brighter. Well, there are a lot of laboratories to equip to keep science moving forward. Provide cutting-edge technology. Invent what science needs. Diagnose people before adaptation. Help customers make their medicine. That makes people better. Over 67,000 strong. Working together for science. To make a difference. To help science advance. Because we all want to live healthier lives. And what brings everyone back to work harder tomorrow is knowing that we make a difference, creating a better place for everyone.
I request the guests, Dr. Shivrakash sir and Dr. Arun Kumar sir, to please switch on their camera. We'll be proceeding for the second session now. Thank you, sir. So without further delay, we'll proceed towards the next scientific session. For that, we are honored to have with us as chairperson, Dr. Shibrakash Ratnam. It gives me immense pleasure to introduce this dynamic personality on this occasion. Dr. Shibrakash Ratnam did his B.Pharm and M.Pharm from Government College of Pharmacy, Bangalore in 1988 and 1990, respectively. Thereafter, he pursued his PhD from LM College of Pharmacy, Ahmedabad. Sir served Zydus and Sun Pharma in various capacities in the area of pharmacology and drug discovery from 1991 for around 7 years. He started his own Codon Research Services Private Limited, a CRO in 1998, which is the first CRO in India in private sector. And at present, he is the founder and managing director of Synchron Research. He also started Intervene, a central lab for clinical trials in 2006, and he is the founder chairman of that company also. Sir also started Advanced Phytotherapies in 2007 for alternate therapies for neglected diseases, and Novel Genetics in 2011, a niche generic company for regulated markets. He was the former vice president of Indian Pharmacological Society and now serving as its president since 2019. He has more than 75 research papers in various international and national journals of repute. Sir has written many articles in various magazines and book chapters. He has also co-authored a popular book on screening methods in pharmacology. Sir is a recipient of Udyog Ratan Award from Institute of Economic Studies in 2008 for his contribution for the development of clinical research in India. With his entrepreneurship skills, Sir has given a huge contribution to the research and development field. We are highly obliged to have you here today, sir. So I thank you for joining with us. And now I request Dr. Ratnam, sir, to please call upon and introduce our second speaker, Dr. Arun Kumar Ramteke, sir, and take over the session's proceedings. Over to you.
Thank you very much. Thank you very much for the introduction. Today I'm really honored, to introduce a well-known person like, Ramteke sir to all of you. He doesn't need any introduction, but as a customer, I need to read out his CV. I must tell you that we are in the middle of the pandemic, and so much of happening in the area of vaccine development and research on the whole spectrum of vaccination of the affected people across the globe. And he is the best person to talk about this subject because of his experience in vaccines and his previous experience in vaccine development. Ramteke sir is a retired senior drug regulatory officer. He was ex-Joint Drugs Controller of India in CDSCO. For about 31 years of experience in drug regulatory aspects in the office of DCGI and CDSCO. He has in-depth knowledge of Indian Drugs and Cosmetics Act, rules and regulations of global regulatory norms, attended numerous workshops, meetings on drug regulatory issues nationally and internationally, including WHO, US FDA, etc. At the time, he was in the helm of the affairs. That is the time where, DCGI really went into great overhaul. So I'm sure, he was in the all good things happening in the DCGI's office. He started his career at Central Research Institute in Kasauli as Deputy Assistant Director, BSQC Division, testing of vaccines and sera as per protocols. He has extensive experience in new drugs, vaccines and biotech products and pharmaceuticals, medical devices, regulatory approvals and development experience. He has also experience in review and evaluation of product dossier for preclinical, toxicological, pharmacological, CMC, quality control, clinical trial data of new drugs, biological and medical devices. Strong skills in quality assurance management, regulatory oversight of clinical trials, and great expertise in GCP, GLP and pharmacovigilance programs, and GCP training and inspection of CROs in India. Currently working as consultant and faculty at Clinical Development Service Agency, CDSA, Department of Biotechnology, Government of India, since April 2012. Involved in training workshops of GCP, Ethics Committee and other regulatory topics all over India since 2013. As an expert involved in 2018 and 2020 online course 1 and 2 on regulatory aspect. I just read his CV, but this is not he. His experience is extensive and he gave great contribution to DCGI's office and his life. He has given his entire life to CDSCO, and none other than Ramteke sir can explain to us the nitty-gritties of regulatory aspects of vaccine development. Sir, over to you.
Yeah. It was so nice of you to explain my CVs. Thankful to you. Nicely explained. And good evening to all the participants and all the people there. and my talk will be the current regulation of the development of biologicals and vaccines. May I have the slides, please?
Yes, we are presenting.
No, this is the last one, last speakers.
Wait a minute, sir. We are doing the next.
Yeah. No problem.
Can you see the presentation now, sir?
Yeah, but it is not clear. It is
So it is
blue.
clear here. Based upon the internet connection, the clarity will vary if we are presenting.
Okay, no problem.
Okay, thank you, sir.
So my topic will be the current regulation of development of biopharmaceutical, including vaccines in India. This is just a disclaimer. As we opening this PowerPoint are intellectual property of mine, and presenter are protected as under the Copyright Acts. So the presentation goes like this. Outline is initially the new drug and clinical trial rules 2019 as per the Gazette notification GSR 227 dated 19th March 2019. Then we can just look into the brief about how to apply, what are the fees, and what are the documents required for approval of vaccines and biopharmaceuticals. There are various guidelines available from for industry that is on this regulation that DBT ICMR also issued us guidelines on biosimilars as well as recombinant DNA product procedure, how to apply, and what is the requirement for DBT for recombinant DNA products introducing in India. So that is the last one. The amendment has been took place in the rules by the Gazette notification GSR 227 dated 19th March. It is called New Drug and Clinical Trial Rules 2019. It is available at the website of the CDSCO. Briefly, this contains the 13 chapters, mainly the definitions and then Ethics Committee for Clinical Trial, BA-based studies, Ethics Committee, Clinical Trial and based new drug procedures, and there is one chapter on compensation. Also, the chapter on manufacture of new drugs and or IND for clinical trial or BA-based studies, its examination and test and license. And also the chapter on import of new drugs and for conducting the clinical trial in India. There are chapters for import of manufacture of new drugs for distribution in India. And chapter 12 is amendment as a rule, as it is this is amendment of the previous is a Schedule Y. Now it is that is removed, but exactly content is the same as per the Schedule Y. So this is just about the brief salient features about the said new rules. We call it NDCT rules 2019, new drugs and clinical trial rules 2019. These rules shall apply to the clinical trials, bioavailability studies, bioequivalence studies of new drug, and regulation of ethics committee relating to the clinical trials and biomedical health research. The main objective of this rule is to promote the clinical trials or clinical research in India, and to have the predictable, transparent, effective regulation for clinical trials. Also to make the faster accessibility of new drugs to the Indian patients, Indian populations. The rules also provide to disposal of clinical trial application by way of approval or rejection or seeking further information within period of 90 days for the drug developed outside India. However, in case of application to clinical trials of a new drug or investigation new drugs as a part of discovery, research, and manufacture in India, the application is to dispose of within a period of 30 days. And the requirement for local clinical trial may be waived for the approval of new drugs if the new drug is approved and marketed in any of the countries mentioned in the specified rules like USA, UK, like that. The name of the countries are mentioned under these rules. Local clinical trial may be waived if it is applicable for this import of new drugs. DCGI has already granted permission to conduct the global clinical trial, which is ongoing in India, and meantime, new drug has been approved for the marketing in the country for specified by the DCGI with certain conditions. There is also the introduction of pre-submission meetings. One can go to the DCGI office and for have the meetings. The pre-submission meeting has been introduced with the fee of rupees 5 lakhs, and also the post-submission meetings. Post-submission meetings also there to discuss with the DCGI office regarding the improve the transparency, efficiency, and predictability of the system. In case of injury during the clinical trial, medical management is to be provided as long as required as per the opinion of the investigator. And there is a formula to determine the quantum of compensation. It is also there. And compensation in case of death or permanent disability or other injury to a local trial subjects that to be decided by the DCGI. Post-trial access approval also given, and there are the expert committees of various group decision which gives approval to the drug or new drug or vaccines. There is also Department of Application for submission of fake documents to the DCGI. So we briefly I just wanted to briefly give the procedure for approval of vaccines in India. Vaccines, as everybody knows, the main objective of vaccines is to generate adequate data on quality, immunity, and or efficacy in the populations. As vaccines are the heterogeneous class of medical product, much of the consideration is for the development should be given product to product basis, and the regulatory requirement may vary depending upon the type of vaccines, whether it is inactivated, live, or attenuated, microorganism base, or antigen base, which is extracted from the pathogens or derived from the rDNA based technology, or by chemical synthesis, like we have the peptide vaccines, or vaccine containing naked nucleic acid, including plasmids for expressing the specific antigen or otherwise. It will also depend on the manufacturing process, its mechanism and action, and the nature of the disease to be prevented, as well as in the target population. So, in general, all vaccines, including vaccines against the coronavirus infection, manufactured or imported in India as required to comply with the requirements of the guidelines specified in the Drug and Cosmetic Rules 1945 and the New Drugs and Clinical Trials 2019. In case manufacture of recombinant DNA derived vaccines, including biopharmaceutical biosimilars, are requirement as per the guidelines described by the Department of Biotechnology, Government of India. So, in brief, recombinant vaccine may be broadly categorized into 3 groups: subunit vaccines, recombinant vaccines. These are the component of pathogenic organism. Subunit vaccine include proteins, peptides, and DNA. Attenuated recombinant vaccines. These are the genetically modified pathogens, organism, bacteria or viruses that are made non-pathogenic and used as a vaccine. Vector recombinant vaccine. These are genetically modified viral vector vaccines used as a vaccines against certain pathogens. Some of the developments made in the production of recombinant vaccines against the certain disease. Biosimilars. What are the biosimilars? Is a biological medicinal product referring to the existing registered product and submitted for the medicinal product registration by the other applicant after the time protection of the data and has been expired of the original product, that is innovator products. Biosimilar product would have abbreviated non-clinical and clinical development program, leveraging on the existing non-information original products and focusing on the demonstration of the similarity within the original products. There are a variety of terms used for the biosimilar products, like in EU, biosimilar products, it's called biosimilar products. In US, it is called follow-on protein products. In Canada, it is called subsequent entry biologicals, SEBs. This is the brief about the regulatory process of biosimilar applications. The review process application includes the locally development of recombinant DNA derived products, initially referred to the Recombinant DNA Advisory Committee. There is a Recombinant DNA Advisory Committee under the DBT, and Internal Biosafety Committee is formed by the manufacturers, local manufacturers. Then the Review Committee of Gene Manipulation, RCGM, at DBT reviews the preclinical toxicological studies, and other recombinant local clinical trial protocol of the, and referred to the DCGI for clinical trial approval. For an applicant, also required to make the application to the DCGI and RCGM simultaneously. DCGI approve the local clinical trials. There are subject expert committee who evaluate the clinical trial protocols and then reports and other preclinical quality and safety data. Based on the satisfactory efficacy, safety, and quality reports, subject expert committee recommends the approval of product to the DCGI. There are various guidelines available for similar biologicals, like regulatory requirements for marketing authorization. It is made by DBT and CDSCO in 2012. Storage of hazardous microorganism, genetically re-engineered organism, or cells notified under the Environmental Protection Act. Recombinant DNA Safety Guidelines 1990 by DBT. Guidelines for generating preclinical and clinical data for rDNA vaccines, diagnostic, and other biologicals. It's also issued by the DBT in 1999. And guidelines for and handbooks for Institution Biosafety Committee is also issued by the DBT. There is a guidelines for preparation of QMS information of new drug approval, biotechnological or biological product. This is also issued by the DCGI, CDSCO. These are the guidelines are available on the similar biologicals. In 2016, it is made available, made by the DBT and CDSCO. Mainly contains these guidelines mainly contains principle for development of similar biologicals, data requirement for preclinical studies, data requirement for clinical trial applications, data requirement for market authorization application, post-market data for similar biologicals, application forms, archiving the data, and retention of samples. Brief about the application for marketing approvals. At CDSCO, there is a portal called Sugam. One can make the application to all new drugs as well as vaccines or all medicinal products, medical devices. All our applications are made under this portal called Sugam, with the fees and all other data requirements. Like there is application form in CT04 for conducting of the clinical trial of new drug, investigation, new drug, vaccines, etc. And data information or data requirement as per the and the table number 1 for new drug and clinical trial rules 2019. Particulars of new drugs. We have to give the name, dosage form, composition, active ingredient, inactive ingredients, indications for which it will be used, manufacturer raw material. This is also the CMC data as per the schedule, second schedule and table 1. One has to give, then animal pharmacology, animal toxicology, then human clinical pharmacology, exploratory clinical trial, phase 1, phase 2, phase 3, or maybe BA study reports has to be given. The clinical trial design generally consists of, this is the template available at the NDCT rule 2019. Investigator and sponsor may design specific protocol as per the investigation product like vaccines, medical devices, etc. They may case-to-case basis. Background information require mainly the scientific and rational data, mainly on the potential on the risk and benefit ratio. So this is just to summarize the design of the clinical trial. Like you have to give the title, short title, which the study, which phases of the study, 1, 2, 3, or 4, investigator, principal investigator, sample size, study population, inclusion exclusion criteria, and the design of the study, which type of study, double blind, mask, placebo control, open label, dose finding studies, etc., etc. Also, you have to give the study duration, start date, end date, name of the product, intervention discipline, description like include dose, duration, frequency, and route of administration. You have to also give the primary objective, secondary objective, and also the endpoints. Along with the protocol, application is required to submit investigator brochure, funding source, ethics committee approval, test license to import or to manufacture the investigation product as per the GMP norms. This is the test license to import a new drug for testing of clinical trial purpose or test purpose. One has to apply in form numbers CT16 with the fees. And when you apply for the clinical trial, that is phase 1, phase 2, phase 3, there are also requirement of the fees. Like for phase 1, you have to pay 3 lakhs, phase 2, 2 lakhs, and phase 3, NDA, 2 lakhs. No fee shall be chargeable in respect of the for conduct of clinical trial for orphan drug, as defined in the clause X approval 2. In case of application received from the micro, small, medium organization enterprises, firm for conduct the clinical trial, approval of new drug, and pre-post submission meetings, the fee shall be payable to half, that is 50%. As I said earlier, there is subject expert group at the DCGI office who evaluate the applications, dossiers of various categories like antimicrobial drugs, analgesic and antiviral, cardiology, dermatology, like that. There are 25 subject expert committee. Vaccine is also there to evaluate all documents regarding the vaccines. Based on the subject expert committee opinion, DCGI approves the vaccines or drug. This is just to give the brief process about the RDNA product. This already we have discussed. These are the guidelines available, and there is a review committee of genetic manipulation, RCGM, under the Department of Biotechnology, is responsible for authorizing the import, export, and for research and development or review and development and review of the data up to preclinical evaluation. After the preclinical evaluation, RCGM recommends the clinical trial to the DCGI. There is also the Genetic Engineering Appraisal Committee. Functions are under the Ministry of Environment and Forest for review and approval of activities involving large-scale use of genetically engineered organisms like LMO and products in the R&D, industrial production as well as environmental release. And of course, there is the Central Drug Standard Control Organization. It is a regulatory body under the Ministry of Health. It is responsible for grant of import, export license, clinical trial approval, and permission to manufacture new drugs, vaccines, biologicals, medical devices, etc. This is the flowchart they have at the DBT RCGM. General approval procedure for recombinant products. Like you have to submit a proposal to the RCGM, then institutional biosafety committee with the DBT nominees, application to RCGM, which gives the laboratory and preclinical data. Based on the preclinical data, RCGM conveys the recommendation to the applicant for conducting the clinical trial. RDSC approves the protocol, and IBFs examine the human clinical trial data, send it to RCGM. GEC approval for environmental release. So, in brief, this is the brief regulation about the vaccines and biopharmaceutical biosimilar products in India by DBT as well as DCGI office. Thanks. If any question, you can just ask.
Thank you very much, sir. It's a very specific, considerably elaborate presentations for designed only for the approval processes for vaccines, and this shows your expertise. That, you spend period of time in your office. That I'm sure you are part and parcel of the whole regulation getting the new phase at your time when you were in the office. And further, there were lot many modifications anyway that keeps on happening. But the group you had in the DCJ's office kickstarted all this, revolutionary guidelines for India. We haven't seen such things before. So all the things happen for good. And thank you so much for sharing your valuable expertise, and that can be seen in your slides. And you have the knowledge of all these guidelines in the back of your hand, so you don't need the slides also. I'm supposed.
I just try to compile with all these requirements.
Yeah. So, is there any questions? Can the moderator ask any questions? Somebody need to, because you will not get such an expert again for your conference. So ask as many questions as you want. He is there to give the clarifications.
Sir, I can see a question in the chat box. Shall I read it to you?
Yeah, please.
As we have heard that vaccines for COVID has been given emergency use, authorization after phase 2 of the clinical trial, and up to phase 2 only safety is established, then why it is being said that the vaccines against COVID are effective?
This question is a little bit tricky. The protocol which was approved by the regulator, it says clinical trial of the COVID vaccine phase 2 / phase 3. And this is not correct that only the phase 2 examine only the safety aspects. Phase 2 also evaluate the efficacy aspects. So efficacy was also examined during the phase 2 and phase 3. This vaccine will continue under the phase 3. But marketing approval has been given because of the emergency situation.
So we have one more question from Anusuya. Do vaccines for animals also follow the same approval route?
No, animal vaccines are different regulation. Like we have the Animal Husbandry Department in the Ministry of Agriculture. They evaluate all the requirements for animal vaccines, animal drugs, animal new drugs. We are not involved in that. We just give the approval.
Dr. Jayant has written, I think Schedule Y is still in force for veterinary medicine. Is it true?
Yeah, it is there. It is there. Correct. He is correct.
We have one question from Dr. Ketan Raj. Will it come under new composition? So he has to apply to Director General CDSCO and Director General Drug Control.
I think he was a little bit confused. CDSO is a central drug standard control organization, which is headed by the Drugs Controller General India.
Mr. Ashish Kaida has asked, safety point of view, which vaccine is better, rDNA based or the one prepared by traditional method? What are the chances of host chromosomal modification with rDNA based vaccines?
All are the different what you call pathways to generate immunogenicity. If vaccine has different efficacy, potency, safety, like mRNA vaccines has now, you heard 90%, 92%, 94% efficacy. So it all depends on the vaccine to vaccine.
Dr. Deepak has asked, but the number of subjects are very limited in phase 2. Then how can we guarantee the efficacy?
How can you say it is limited to the? How can you say, because they have inoculated in the number of population, and the reports has been more than 1,000. And it is still continuing.
If our chairperson allows, we shall conclude this session, sir.
Sir, I take this opportunity because you are here. What is your view on considering this pandemic situation and the controversies surrounding this vaccine development, regulatory processes, and considering our own self-sufficiency in discovering new vaccines for such emergency situations? So what should be our preparedness as a nation, in your opinion? And the second question would be, we have ACC members, so should we not make ACC members public, like who are the people, the type of scientists, and the intellectuals who are involved in ACC approvals of such vaccines? Can't we make the ACC members' names public?
Yeah. Your last question, I said it is I think it is on the website of the Controller General. There are name of the different SSCs, subcommittees. There are 25 subcommittees are there. And sometimes back, the list was on the website. But it has, well, they changes frequently. Some experts may change, some experts may leave, like that. So it's good. Sometimes it is they withdraw the list, and then again the fresh list uploaded, because it has to go a little bit procedure approval from the ministry, and then uploaded the names of the experts. So sometimes it takes time. So there is the procedure. Names are available on the website. And regarding the vaccines approval is strictly as per the regulations, rules prevailing in the country. There is no compromise on the efficacy, safety, and quality of the vaccines. All are vaccines.
Thank you very much, sir. It was really enlightening to have you here. I feel as though you are still in the office, not retired.
No, I'm at home now.
Thank you very much.
Thanks.
It was a very enlightening experience, to listen to you and to share your experience with us, and to the audience and the people who have participated to hear you. And I, on behalf of LMCP and all the LMCP team members, I thank you very much, and thanks for coming and to share your experience with us.
Thank you, sir. Thank you very much.
We once again thank you.
Thank you.
Thank you, Dr. Ram. Take it.
Thank you, ma'am.
Thank you, Dr. Shiv.
So please be with us.
Yeah,
Please be with us.
Thank you so much
Thanks.
for joining.
We once again thank Dr. Ramteke sir for such an informative and interactive talk, and also for sparing his valuable time to be a part of our event today. And I request Dr. Ratnam sir to please present a certificate to Dr. Arun Kumar Ramteke sir as a token of our love and appreciation.
Sir, kindly accept the certificate as a token of our appreciation.
Thank you, Dr. Ratnam. Thank you very much. Thank you very much. Thank you for all LM team.
Thank you, sir. Thank you very much, sir.
Yeah. Thank you very much.
Thank you, sir. And we are very thankful to you also, Dr. Ratnam sir, for chairing the session in a very nice way. And I request you to please accept the certificate of appreciation as a token of our gratitude, Dr. Ratnam sir.
Thank you very much. Thank you very much. Thank you very much to LMCP, and as well as to the LMCP team, all of you. And I also thank Thermo Fisher for forthcoming sponsoring such wonderful conferences. Thank you very much.
Thank you all. We'll be starting the last session in a few minutes due to some technical glitch. So please be with us, participants. And for the next session, I'll be inviting Dr. Palak Parikh to coordinate it. So be with us, participants, please.
रामटेके होता
Dr. Arun sir, you may leave the session now
Yeah.
by clicking the cross button of your browser.
Okay. Yeah.
Okay. Thank you so much, sir.
Thanks.
The people of Fisher Scientific, start every day to help move science forward.
So we can all live healthier lives
On a cleaner planet.
for a safer world. This is why we work.
To help solve. To save. To advance chemistry. Because when we partner with science,
We make the future brighter. Because there are a lot of laboratories to equip to keep science moving forward.
provide cutting-edge technology. Invent what science needs.
Diagnose people before they are patients.
Help customers make their medicine.
That makes people better. Over 67,000 strong.
Working together for science.
To make a difference.
To help science advance.
Because we all want to live healthier lives.
And what brings everyone back to work harder tomorrow is knowing that we make a difference, creating a better place for everyone.
Good evening, everyone. May I now request you to please switch on your video. Moving towards the next scientific session, we are honored and privileged to have with us Dr. Chhavi Agrawal Madam as a chairperson with us. And it is an indeed privilege for me to introduce her. Dr. Chhavi is currently working as the product marketing manager at Thermo Fisher Scientific. Madam has completed her PhD from Jagiellonian University, Poland on membrane protein signaling. At Thermo Fisher Scientific, she is responsible for marketing activities for cell biology and bioproduction workflows. We feel honored and privileged to have you here today, Madam, and extend a warm welcome to you, Madam. May I now invite Dr. Chhavi Madam to please introduce our third speaker, Dr. Alejandro Barreca, sir, and take over the session proceedings. Over to you, Chhavi Madam.
Thank you so much. My audio is clear. Can you confirm me once?
Yes, Madam, pretty much clear.
Okay. So thank you to the organizers for organizing this event and giving Thermo Fisher Scientific an opportunity to be part of it. I would like to invite Dr. Alejandro Becerra. He is the principal application scientist for the Global Purification technical lead. And he has been with Thermo Fisher with 14 years of experience in downstream processing, with a focus on chromatography applications for the purification of monoclonal antibodies, AAV, and other biotherapeutics. Alejandro holds a PhD in chemical engineering from Cornell University, and today he will be helping us understand the supporting developments of mRNA-based therapies by addressing the large-scale purification challenges. So I would request Dr. Alejandro, if you can share your slides with the slide option on the right side. You can share the slides from there.
Yeah. trying to work through it. Sorry.
Yes. Once you press these slides, you have to give add a new presentation. Just give a random name to the presentation, and
Okay.
then you can upload it. Might take a few minutes.
Yes, it's working. Thank you. Okay, it's uploaded, so.
Okay, over to you then.
Can you see my screen?
I can. Can organizers confirm if it is visible on the audience screen?
Yes, madam, it's visible.
Okay.
Okay. Thank you very much. Apologies for that small technical difficulty. And again, thank you for allowing us to share some of our experience here and our interest in supporting mRNA-based therapy. So I will talk to you today about a few different topics in this area. I'll put first things into context with a little bit of an introduction, then focusing on a specific solution that we have developed for this application, an affinity chromatography product. And then I will move on to sort of a case study and feasibility to demonstrate the potential use of these tools with some data. And lastly, I will briefly touch upon some of the analytics that could be used in the field and for which Thermo Fisher has some options. So I think today, in the middle of a pandemic, I think mRNA applications have come to light, and particularly in the context of a vaccine, right? So that's one of the main applications for this technology, but it's not the only one. Certainly, the proteins that are encoded into that mRNA can have different functions. For instance, those can encode antibodies to be expressed and secreted into a specific cell type. These mRNAs can also encode for gene editing tools like CRISPR, and those can be utilized as well with this technology. And similarly, these mRNAs can also code for proteins that will provide some signals to the cells and, for instance, induce them for certain types of differentiation. So there's a lot of opportunities and applications. So it's quite diverse, the field here. And the use of these technologies is not only, as we see today with the COVID vaccines, namely with an in vivo application, but as I mentioned, maybe for some of the gene editing tools, this technology can also be used ex vivo in the context of cell therapy. So there's certainly a lot of opportunity there, and again, we've seen, fortunately, a quick uptake of the mRNA in the context of vaccines, but this is likely just the beginning of this type of therapeutic. I think it's expected to have a significant growth, just like we saw maybe 10, 20 years ago with monoclonal antibodies, and something we're already seeing with other forms of gene therapies, particularly with virus vectors like AAV. So I think it's likely that we'll see that increase. And with that in mind, we need to have the right tools to be able to support the delivery and growth of those technologies. And although these mRNA therapeutics are relatively new as an approved therapeutic, again, with the example of the COVID vaccines, the science obviously dates back around 30 years. So in the 90s, some of the initial science that was developed was proven with some trials in animals as well, and with ex vivo applications. And then throughout the 2000s, now we saw some of the actual first clinical trials and using these mRNA therapeutics in humans. And again, just with the coming of the COVID vaccines, I think we will only see many more of these, both again in the context of vaccines, but also for other applications. And I'm not going to dive into the manufacturing of the mRNA, namely the in vitro transcription reaction, but it's important to put it into context. What are the, at least at the general level, the steps that are used in the synthesis, because then from a purification perspective, we need to know what's coming into the process that needs to be removed to deliver and have a safe product at the end that can be delivered to patients. So generally speaking, the manufacturing entails 3 different parts. The first one being just the synthesis of the mRNA, and then the purification, and lastly, the delivery, usually with lipid nanoparticles. In the context of the synthesis, the first part that is required is a DNA template. Today, for the most part, that generally is based with the use of plasmids, and within those, Thermo Fisher does have some tools that could be utilized for that, both development and production. And then really at the heart of the manufacturing is the in vitro transcription reaction, where we take this template and then we utilize the building blocks of the mRNA along with some enzymes, not only for the transcription, but also some accessory enzymes that will ensure the reaction goes in an optimal way. And that's really the key, this second part here, the second step. The third part that is listed here as RNA synthesis is not really directly related to mRNA, but just in general, other types of RNA, for instance, RNAi or just other oligonucleotides, can also be used for similar type of applications. And for those, we also offer some tools at Thermo Fisher. But again, today the focus will be primarily on the purification. So once we have completed that IVT reaction, we need to be able to purify that mRNA. And lastly, I will not cover it either, but once that mRNA is purified, then it's to be put into the right delivery vehicle that is generally a lipid nanoparticle. And to that end, Thermo Fisher also offers some products. But again, today I will focus mainly on the purification part here, highlighted in red. So, as it's a relatively new application in the context of a scalable GMP process, there aren't many options that are scalable for the purification of the mRNA. Here I'm listing a couple of options. Some that are more commonly used at the lab scale are not listed here, but those include lithium chloride precipitation, as well as some extraction and precipitation with organics, or for instance, the utilization of some kits that utilize a silica column essentially for the purification. But those are really not easily scalable, right? So extraction and precipitation are not used for the production of other biologics. The ones I have listed here are some other potential options that could potentially be implemented, but most of them have some disadvantages, particularly with regards to the selectivity, as well as the scalability and the potential capacity that is critical for any chromatography process. Others, like size exclusion chromatography, while they have been used and scaled up with older processes for other biologics, also have some great limitations in terms of the amount that can be loaded and the efficiency of the separation. And last, what I'm listing here is the potential option of utilizing affinity as a tool, and that certainly has many advantages. I guess the only disadvantage is it will not be able to distinguish between some of the product-related impurities, but generally speaking, that will be the tool that will be more easily scalable for this type of application. And that will be the focus of the next part of the talk. So why do we look at affinity as an option? Certainly, the most well-known and utilized affinity capture step is protein A in the context of a monoclonal antibody, and the advantages it provides are several. First, in a single step, you can obtain a high purity and yield, and it makes it easier to have a similar process that can be platform for different molecules. In the context of mRNA, the load material is certainly not as complex as a clarified cell culture, but still the ability to provide a platform process as well as still providing high purity and yield with a single step and minimal process development is still one key advantage. And generally speaking, it could be able to also reduce the number of steps. So on the options that I listed previously, it's likely that one will need to utilize more than one step, potentially up to 3, with the use of affinity. Whether it's in biologics or specifically on mRNA, there's always the potential to reduce those steps. And by doing so, the overall process yield can be increased. That's what's illustrated with the graph in the middle. So specifically for mRNA, we're not talking about an affinity resin that is an antibody, but we're leveraging the interaction between the base pairs, specifically the characteristics of the mRNA, that it contains a poly-A tail. So we can utilize that as an anchor, and with the use of a ligand that consists of a poly-T, a polydeoxythymidine, we can take advantage of that hybridization or base pairing between those two to come up with a solution that utilizes and selectively purifies the mRNA. It also has other advantages in the context that it can be utilized just at a high ionic strength, so relatively simple, and the elution can be performed in water, but I will go into some examples of that later on. One key characteristic of any chromatography, or one requirement, I should say, is that it needs to have relatively good capacity to make it cost efficient. And for these larger molecules, depending on the size of the construct for the mRNA, we have seen what we will consider very good capacities on par or higher with other technologies. Here I'm just listing an example with a 5 mg/ml binding capacity, but I will show some more in-depth data later on. And because this is what I'm talking about here is a chromatography resin, the scalability of this type of material is well documented as used for the production of all kinds of biologics. Specifically, in our case, we're utilizing the porous base bit for this particular affinity resin, and it has some inherent physical characteristics that provide some advantages. The first one, because the material of construction is polyvinyl divinyl benzyl, so a polymer, it makes it a very rigid material. What that means in the context of purification is that the relationship between pressure and flow is linear and irrespective of column diameter, which I'm sorry, was there a question? Maybe not. So going back to the characteristics of the permeability is quite good, so a relatively low pressure drop, but more importantly, is easily scalable. The second characteristic that we utilize in the making of this affinity product is the relatively large pore sizes of the porous beads. So compared to other materials, the pore sizes are larger. Specifically for this resin, the pore size of the base bead is about 200 nanometers or 2000 angstroms. And lastly, in the context of chromatography, there's always a balance between the particle size and the pressure drop. But again, because it's a rigid material, we utilize a 50 micron bead that will increase the overall surface area in the column compared to a larger size. But we can still do that while keeping an acceptable pressure drop across the bed. So those are some of the reasons we chose that particular base bead for this. And the animation is not working here, but how do we use it in the context of the separation? It can be used in two ways. The first one is just using it, as I have been describing, as a capture step after the IVT reaction that we will purify, in principle, only mRNA that has a poly-A tail. There can be product-related species that also have the poly-A tail, which then you may be able to remove with a second step that is listed here as potentially hydrophobic interaction, an ion exchange, or reverse phase. The second option, it's not clear from the slide, and I apologize for that, but it will be to use it as a second step. So there's some initial cases where maybe it's found more useful to do some other type of purification with chromatography first, and then follow that with the oligo DT resin as an affinity, so almost as a polish step. Just to clarify, I'm not suggesting to use the resin twice in the same process. It's just to utilize it either as a capture or as a polish step. So with that in mind, we actually were able to collaborate with some partners and obtain some mRNA to further understand the performance of this material and do a little bit of process optimization. Maybe not as in depth as it will be done for a process, but at least give us a better understanding of the characteristics of this resin. And that's what I'm going to focus on next. Also guide you in case you start working with this material, what could be some of the steps that could be performed to get to an optimized process. As I mentioned before, we actually load the material at high ionic strength. The reason for that is to mitigate the repulsion between the mRNA and the oligo DT, such that a hydrogen bond can be formed between the two, allowing for that hybridization. Now, when we add a high ionic strength solution to the mRNA, there's always a potential to precipitate some of the material. So the first experiment we conducted was to evaluate that potential precipitation with two different types of salts, in this case, sodium chloride and potassium chloride. And we actually had the opportunity to test this with 3 different mRNAs that differ in length, going from 1,000 to 3,000 nucleotides in length. So to assess whether there was any precipitation, we only measured the absorbance at 600 nanometers. What we see on the graph on the left is there is really no significant change in terms of the absorbance for 2 of the constructs. However, we do see an increase in that absorbance for the 2,000 nucleotide mRNA. So that tells us that if we go to that concentration of sodium chloride, there will be some precipitation. So if we want to load these resins, we need to stay at a concentration below that. We perform a similar experiment, but now in the presence of potassium chloride, and we can see that there isn't that same increase in absorbance with the 2,000 nanometer mRNA. However, we do see a small increase for the 1,000 nucleotide construct at around 1.8 molar. So again, this only provides us the maximum salt that we could utilize, and that will be sort of a first step to understand that, generally speaking, we may not need to go to those high salt concentrations, but it could be beneficial to improve the binding capacity. And that is what I'm showing here next. So this data I'm presenting here is just conducted in a 96-well plate in a static mode. So what we looked at is different concentrations of sodium chloride as well as different pH levels for the same 3 different constructs, again, 1,000, 2,000, and 3,000 nucleotides. And what we determine, essentially a mass balance, determine how much of the mRNA bound to the resin. In some cases, all of the mRNA bound, so we cannot really call this a static binding capacity, but it's getting us in that direction of what will be the binding capacity. Generally speaking, for instance, if we take the figure on the far left, we can see that as we increase the concentration of sodium chloride, we see a small increase in binding capacity. That is not surprising, again, since we are further mitigating the repulsion between the mRNA and the oligo-DT. We see a very minimal impact of the pH within the area that we evaluated, and a similar trend for the 3 different constructs in terms of the sodium chloride concentration, just different levels and different levels for the binding capacity because of the size. So the optimal conditions will differ, but again, this still allows us to find some conditions that then we could utilize in a column format, right? So the final goal of this is to put this into a process with a column. And so the next thing we need to do is actually evaluate what is the actual binding capacity of these resins. And to do so, what we have to do is generate a breakthrough curve, and then we can determine the binding capacity. In this case, we performed that test with 3 different concentrations of mRNA in the load material, and specifically with the construct that was 3,000 nucleotides, and we determined the binding capacity at a 5% breakthrough. In the next slide, I will show you some of the breakthrough curves, but focusing here with this set of data, again, these molecules are larger than most proteins, so there is still some mass transfer resistance, which means that if you increase the residence time, you will increase the binding capacity, and we can observe that trend for the 3 different load materials. But the other part that we can take from this data is the influence of the load concentration onto the binding capacity. So if we take a step back, the binding capacity is driven by diffusion, and diffusion in turn is a function of a concentration gradient between the mobile phase and the stationary phase. So it's not surprising that as we increase the concentration in the load material, there is a higher driving force that in turn results in a higher binding capacity. So for example, if I look at the 2-minute residence time data, we can see an increase in binding capacity with the green data point compared to the blue one, just because of the concentration. So just understanding what are the parameters that are important during the development of these processes. Now, if we look more closely at the actual breakthrough curves from which we determine the binding capacity, this is what we're seeing here on the figure on the left. And we performed these experiments for the 3 different constructs, and we can see a couple of things. One is that the binding capacity, as I mentioned, is greater than 2 mg per ml for most cases, but the breakthrough is relatively shallow, which means that you could potentially load to pass the initial breakthrough without sacrificing much material in the load flow through. So, for example, on the figure on the right, what we did was load to 2 mg per ml, which we can see there's a little bit of breakthrough for the 3 different constructs, but even under those conditions, we get over 90% recovery for the 3 different constructs. So again, just an example, in principle, we could load less and potentially improve the recovery, but understanding how these breakthrough curves look will also help us with the development of the process. So all the data up to now had been generated with pure mRNA, but again, in the context of the actual process, the load material is going to come from an IVT reaction. So to confirm the results we had observed before, we actually took load material coming from an IVT mixture with the conditions that we had found that obtain good capacity. And what I'm showing here is just a chromatogram. What we see on the far left is during the loading of the sample. Most of the other impurities present in the IVT mixture will flow through, right? Because they don't have a poly-A tail, and those include the DNA template, residual enzymes primarily. Then we can wash those off, and what you can see in the area where it's labeled wash 2, what we can do is reduce the ionic strength without having an elution, but potentially removing some additional impurities. And then the elution can be performed with a very low conductivity buffer or water in this case. And you can see a nice and sharp elution peak that will contain only the mRNA that has a poly-A tail, which is primarily single-stranded DNA, and there could be also some double-stranded mRNA. Sorry, I said DNA, it's mRNA. And just like with any chromatography step, after dilution, we will clean the resin so it's ready to be reused again. So looking at the performance, both in terms of recoverability and purity. So what I'm showing here first on the left is when we conducted this experiment with the actual load material from the IVT mixture, we observed the same recovery as when we performed the experiment just with a pure mRNA. So as expected, with an affinity material, it's only binding the target product, or primarily the target product. And then on the right, we assess the purity just in terms of the protein content. Again, these IVT reactions have all these different enzymes. So what we measured was just the amount of protein in the load material. You can see most of that protein is found in the flow-through. There is nothing in dilution. And the rest we can actually find in the cleaning fraction, just that difference between the load and the flow through. So a very clean material in terms of protein in dilution. We further analyzed that dilution fraction and compared it to what I mentioned at the beginning at lab scale, maybe a more common method to purify mRNA, meaning a silica spin column. So what I'm showing here is chromatograms from a reverse phase analytical column that allows us to characterize further the samples. So on the top chromatogram, what we see is the actual mixture. The peak at the very beginning of the chromatogram is all these enzymes that I've been referring to, so all these other impurities. And then the target mRNA is further to the right on the chromatogram. When we compare it to the spin column format purification and with the porous oligo-DT resin, we can see the same peak, roughly around 8-minute retention time. But once we zoom in into that area for that mRNA peak, we can see there is this second shoulder, sort of a pre-peak, but that is actually higher in the load material and even on the spin column elution. And we're still working on understanding the characteristics of that material. We suspect is product-related species, meaning mRNA that does have the poly-A tail, but may have a truncated sequence. But even comparing it to the silica purification, the percentage of that peak is smaller with the oligo-DT resin, and potentially, as I mentioned at the beginning, you could have a secondary polishing step to remove those. Now, with any type of resin, and particularly affinity resins, it's important to ensure that they can be reused, right? We don't want just, this is not a silica kit, right? We're not going to throw these things away. We need to be able to reuse them. So one experiment we conducted to make an initial assessment was to see if there was any carryover of product after repeated cycles. I think the data here is, some data here is missing, but essentially we looked at two things: whether there was any carryover of product in terms of UV when performing a blank cycle after these 10 runs, and that is what is shown on the left, where there is no carryover in that elution fraction with a blank run. And then on the right, I apologize, I don't know what happened here, but essentially we didn't see a change in recovery. In all cases, it was above 90% throughout those 10 cycles. And with any resin, the other important characteristics is just to be able to clean the material. And to do so, what we conducted was an stability study in the presence of both acidic and alkali conditions with hydrochloric acid and up to half molar sodium hydroxide. Although this is an affinity resin, because it has this oligonucleotide ligand, it does have some ionic capacity, so somewhat similar to an ion exchanger. So we utilize that measure as a surrogate of the stability. And what we see is that even after 2 days in contact with one of these three solutions, we really don't see a difference in terms of the ionic capacity, suggesting that you can utilize these cleaning solutions for removal of tightly bound impurities, as well as for sanitization of the resin. And last, I just wanted to point out that within Thermo Fisher, in other parts of the organization, we do have some other tools that will allow us to characterize these mRNA therapeutics or tools better. For example, again, I didn't go into the details of the IVT reaction, but another important aspect that is required is the capping of that mRNA. So there's a cap at the 5' of the mRNA, and to be able to determine the efficiency of that capping, it's important to have the right analytical tool. To do so, one can utilize reverse phase. So an example is shown here at the top, and that can be coupled with mass spectrometry to further understand that part. And actually, that's some of the ongoing work on the example I showed before to understand what's present in that pre-peak. Similarly, one also needs to understand the purity. So that's directly related to the example I was showing, whether there is potentially double-stranded RNA or incomplete transcripts, and the same kind of tools can be used for that purpose, namely that reverse phase and mass spectrometry. And lastly, the actual identity of the mRNA can also be confirmed with similar tools. So Thermo Fisher, in a separate division than ours, has tools appropriate for those analyses. And with that, I want to thank you again for your time and allowing us to share some of our experiences here. And I will open the floor for questions.
Thank you, Leandro. Can organizers help me with the questions, because I had to refresh my browser?
Yeah, sure.
So I did not see the questions. Thank you.
We are having a question from Nirbhay Gandhi. What is your view for future of mRNA vaccine in third world countries like India?
I think I hope these type of therapeutics can be and the technologies can be transferred to different countries. I think we're already seeing some of that, right? I think one of the few positives of the pandemic is that the technology transfer of these vaccines has started from the originators. So maybe that will kickstart some of the knowledge transfer into these other regions. And again, with this exciting time, I think it's possible that some companies may also start their own businesses in those regions. So hopefully we'll see a wider availability of this type of therapeutic.
Dr. Jayant has asked, sir, can you say something more about current therapeutics other than vaccines for mRNA-based products?
Yeah, I think obviously the key players in the news are Moderna and BioNTech, right? But there's others, and even just looking at the pipelines of those two, you can see that there's not only the vaccines for other types of pathogens, but some that I mentioned at the beginning was utilizing this as a tool for gene editing, utilizing CRISPR or things like that in an ex vivo setting, but also potentially having the mRNA expressed antibodies, right? So that's something else that is being investigated, and some of this information with regards to the pipelines for these are publicly available, right, for those bigger companies. So that's another source for the audience to find more details.
So Dr. Ketan has asked, what is the difference between purification of simple mRNA vaccine and liposomal vaccine?
So the mRNA, so we make the mRNA first, right, with the in vitro transcription. So that has to be purified. And then the lipid nanoparticle is separate, right? And then the mRNA has to be put or encapsulated into the lipid nanoparticle. So they go hand in hand. So I really don't know much about the lipid nanoparticle purification per se, but once you have the lipid nanoparticle, then essentially you need to encapsulate the mRNA. So you cannot just have the lipid nanoparticle or the mRNA. One is the delivery vehicle for the other.
Okay, sir. He has also asked how to remove the multivalent metal cations.
So on the affinity resin that I presented, those should not be present. There are some salts that are useful to help with the stability of the mRNA, but the things that are unwanted can be removed through the chromatography step, and I didn't show it here mainly because Thermo Fisher doesn't have products for that. But just like with any other biologic, after a chromatography step and prior to the encapsulation to the lipid nanoparticle, one performs a buffer exchange. So TFF could be another option where any residual ions or unwanted salts could be removed. So for example, for these vaccines, generally that final formulation product contains some sugars for the stability, again, prior to the encapsulation into the lipid nanoparticle.
So we are not having any more
So,
questions. I request,
can
ma'am.
I ask you a separate question other than the therapeutics? Can we involve you people like Thermo Fisher in coming out with small workshops to so that our students get more knowledge in separate workshops, like 2-day, 1-day, 3-day workshops on biologics?
I'll leave that question to my colleagues to answer.
Okay, then let Dr.
Yeah, I'll just add to that that would be great for us if we could be part of the workshops and help the future, let's say, biopharma people to understand the latest technologies and innovation, which is currently prevalent in the industries, and where Thermo Fisher is part of it and can help them in future if they require any support. So that would be great for us. So in case the LMCP requires that, we would be happy to be part of it.
Sure, we'll get in touch with you. Thank you very much, Dr. Yandro and Dr. Chavi.
Thank you again for having.
Yeah, see you.
We once again thank Dr. Becerra for enlightening us with his knowledge and expertise, and also for sparing his valuable time for our event. May I now request Dr. Chavi to please present the certificate to Dr. Becerra as a token of our gratitude.
So thank you, Dr. Alejandro. Great session, and thank you to the organizers, LMCP, for organizing a really amazing event. I have heard of some of the sessions, and they're really very fruitful, actually.
Thank you.
Thank you. Thank you. Happy to help.
I'm sorry. Please continue, sir.
No, I was just thanking the organizers again. Thank you, Chavi, and just happy to help with what we can.
You're always welcome, sir.
We are very grateful to you also, Dr. Chavi, for chairing the session in an excellent way. And ma'am, I request you to please accept the certificate of appreciation as a token of our love and gratitude. Thank you so much.
Thank you so much. Thank you so much.
Yeah. Welcome.
So we are moving towards the end of the sessions. There are some announcements for the participant. The link for the quiz and the feedback of today's session has been provided to you in your email ID. The link will remain active till 12 noon tomorrow. So please fill it before the deadline. Based on that, your attendance for the event will be considered for today. And please also note that completing the feedback and quiz of all the 4 days is compulsory for you to receive your e-certificate. So with this, we have successfully completed the day 2 of our international conference. We once again offer our sincere thanks to all the chairpersons and speakers of today's scientific sessions. We are extremely grateful and thankful to GSPTM and Thermo Fisher Scientific, our industry partner, for sponsoring this event. I thank all the delegates for their keen participation and kind cooperation during today's event. I'm sure everybody has gained something from the conference in these 2 days, and I assure you that there will be more to come. So do join us tomorrow again for the third day of the e-conference at 4 p.m. sharp with the same participants link that you received today. So once again, thank you all and see you tomorrow. Good night. Thank you so much, ma'am. Yamini, ma'am.
Yeah, welcome. Thank you to you too.
Yeah, we'll
Bye.
stop airing the meeting now, ma'am.
Yeah.
Okay.